cell. So no more new discoveries had been made. At the same time, what actually had to be explained is turning out to be more and more and more incredibly complex. So actually, nothing has happened. It's just, there was this kind of, glimmer of hope in the 54, and since then, practically nothing has happened. That was the gist of this chapter by Simon Conley Morris in his book in 2003. And in one sense, it's no wonder if you actually look at, say, amino acids, and if you know what amino acids are, that also puts everything in perspective and actually shows that probably it was, how the experiment was publicized in 1954 was basically hype. Because yes, it's true that proteins are made of amino acids, and cells, to a large degree, are made out of proteins. But still, the jump from one amino acid to a functional protein is incredibly vast. It's such a difference in complexity. Like the difference, it would be something like if someone was in Egypt and by chance, doing some excavation, he stumbled upon a stone block, naturally formed square stone block. You see, this is actually formed naturally, these square stone blocks. And then you publicize, now I know how the pyramids they were built. It's actually, it's just because you have a building block doesn't mean you know how the building was made. There's much more to, to a building than just knowing how building blocks are formed. And so that's actually kind of the proportion between an amino acid and protein. Amino acids, I say here, I had here on Wikipedia, they have, of course, an entry on amino acids. And there are different kinds of amino acids. You see up to the right here. That's up there. That's a simple amino acid. To the left, there is actually the N means nitrogen. So typical for amino acids is they have this 2 hydrogen, 1 nitrogen connected with a hydrogen carbon, and then there's some variation on the others, and that creates different kinds of amino acids. You see, and actually, you can make hundreds and thousands of different amino acids, and 20 of them they are relevant for biological life. 20 different amino acids. They are what actually are. Proteins are made of 10, 20 different amino acids, assembled in long chains. A protein is a long chain of amino acids, and and actually an average protein in a cell is something like 300 units, 300 amino acids long, very precisely organized. And a regular cell needs at least, the most simple cell at least need 200 of such complex proteins to function. So that's kind of, you get the idea, the difference between an amino acid and an actual protein. And and so basically that's where they were able to create simple amino acids and also some other simple chemicals that are compounds in the other complexities of life. But besides that, they haven't been able to demonstrate or make models of how more complex proteins could arise, for example. If you look at it a little more closely, we get an idea about actually the difficulties that have to be surmounted for a spontaneous process create complex proteins. And we can actually appreciate that actually, I mean, even this is not going to happen in the future either. We're simply talking about some thresholds which cannot be, which natural processes cannot get over. For example, what we're looking here now is at some of the different some of those are actually many reasons why these whole scenarios of life arising gradually, such as these apparent hypotheses of life arising from the primordial soup over hundreds of millions of years. Actually, it's we're talking about some completely impossible scenarios. One thing is, the first problem is that Stanley Miller, and also there have been many similar experiments ever since his time, he and other scientists are assuming, and apparently was assuming, that the early atmosphere on the Earth was what they call reducing. And in a more simple way to say that it's reducing is simply it's an atmosphere that has no oxygen. It's an atmosphere without any oxygen. And that seems to be a necessary condition for any process to start going, because oxygen interferes with all other processes. And oxygen is causing fire, is an oxygen process. Rust is an oxygen process. Free oxygen, like O2, is a very active chemical, and it interferes with all kinds of reactions. So if, for example, if you had in Miller's his experiment, if you had oxygen, you know what would have happened? It just exploded. Like methane and oxygen. Methane is the gas you get out. You cook with methane. It's gas. So there's no oxygen, then it cannot burn. But if you had oxygen, then the whole thing would explode. So it seems to be a necessary condition that there was no oxygen on the Earth. And many assumed that that's granted because they think that oxygen has arisen when plants arose on the Earth and photosynthesis started producing oxygen. So that's true. I mean, that's probably the major source of oxygen on the planet today. But geophysicists have discovered also that this condition probably was not true, that actually there was oxygen on the early Earth. Perhaps not to the degree that we find it now, but still to a significant degree, because there are other processes which are also producing free oxygen, such as ultraviolet light, what's called dividing or dissociating water into hydrogen and oxygen. I think it's called photodissociation. So that's a process that also generates oxygen. And actually it has been proven by studies of rocks from the early Earth. From the very, the Earth is today they think that the Earth is something like 4.5 billion years old. And rocks from the very early Earth, also there are traces of oxidation. I mean, in other words, kind of rust, or other like similar process is also found on the early rocks before there was supposed to be life on this planet. So there actually seems to have been oxygen on the early Earth, which immediately, nullifies and stops all these processes that these ordinary life researchers are proposing. So that's one thing. It seems that Stanley Miller, he did not have a realistic atmosphere for the early Earth, neither did Oparin. assume. I mean, it was a different atmosphere. It had free oxygen and it had no hydrogen also. They assumed there's hydrogen on the early, free hydrogen, but probably there was no free hydrogen. There was hydrogen bound as water, but not free hydrogen, because hydrogen is an extremely light element. It's the first of all the elements, and it's extremely light. So whatever free hydrogen is there would actually escape out into outer space. The Earth's gravity would not be able to hold free hydrogen. So for that reason, there was probably oxygen and there was probably no hydrogen. The whole atmosphere that Stanley Miller was proposing was actually not at all realistic. So that's the difficulty number one. From the very beginning, we talk about something unrealistic, not a. Then there's another problem in the experiments that we had at Miller's experiments, was that when Miller was conducting his experiment, he would isolate, during the experiment, the substances that were created. He would isolate them for later analysis. Simply pull them out of the reaction that was going on. They would collect different substances, would collect on the bottom. He would pull them out for later analysis. However, that kind of isolation of desired substances does not happen in nature. They remain as part of the primordial soup, and they keep reacting. So it seems that even if they were made some favorable compounds were made, they would almost immediately again react with other chemicals and be broken down and like that. So they wouldn't actually to accumulate favorable compounds such as amino acids. It's hard to see how that actually could happen. So that's the next problem. How does nature concentrate and isolate the needed elements? So that's then, and furthermore, there are also all kinds of other cross reactions taking place, which are disturbing this, the creation of the needed amino acids and other necessary compounds for organic life. Then, let's see here. Okay. Also, one difficulty that's been mentioned here is just like geologically. I mean, if there had been a primordial soup, then it would be possible to also find traces of it in geological settlements, which are from before life should have arisen. But there's no trace of such a prebiotic or primordial soup anywhere. And if it had been there, actually, geologists would have found traces of it, but there's no traces of such a primordial soup. That's one difficulty that's discussed here. And then, then there is a further difficulty. I mean, just to get to the point where you actually get amino acids, the right kind of amino acids in sufficient quantity, seems like to be insurmountable difficulty. But let's assume it actually happens. Somehow or other you got quite a number of amino acids. But then actually there's one difficulty that immediately arises. Amino acids come in two varieties. They have of each amino acid, it has exactly the same chemical formula. Still it's two varieties, like the left hand and the right hand. Actually it's completely the same anatomical structure, but they are the mirror images of each other. So similar amino acids is acids, chemicals that actually come in two varieties, left-handed and right-handed, they're called. And in nature, when amino acids are generated spontaneously, you get 50% of each. But in living organisms, in living organisms in our cells, there are only left-handed amino acids. There's no right-handed amino acids. Right-handed amino acids are like poison for organisms. They only use left-handed amino acids, 100% left-handed amino acids. So that's the next great issue. Where is a natural process that only selects left-handed amino acids and not the right-handed amino acids? To date, no one has discovered such a process that sorts, only selects one kind of amino acid, only generating one kind of amino acids, not the other kind of amino acids. So, and this discovery actually is again, it's quite old discovery. It was actually discovered by Louis Pasteur, French, you know him. Yeah, he's I think actually next to Darwin, he might be the most famous scientist. Because everyone, when we drink milk, you always drink pasteurized milk. So that comes from him.
Do you know where he is born?
Huh?
Do you know where he is born?
France?
Yeah, in Dole. I've
Dole?
been there. Dole.
Okay, good, yeah.
So since Prabhupada says milk is good for understanding higher subjects, that actually is contributing to the Hare Krishna movement.
Pasteur.
Pasteur.
Yeah, and actually, Prabhupada, said life comes from life. That's actually a quote from Pasteur. Pasteur, he said that because Pasteur, he was an anti-Darwinist. He was really against Darwin. But as a scientist, he said this doesn't. We only see that life comes from living, from what was already living, never from dead matter. And he would, he was very outspoken against and Darwinism. And he made experiments that actually demonstrated that this idea that life can arise from dead matter, from chemicals, it doesn't happen. He was one of those who killed this idea about the spontaneous generation of life. Some different experiments. Pasteur was quite a great personality, very religious, and very excellent scientist, great scientist. So, and he also discovered around 1880 that amino acids, they come in these two varieties, left-handed and the right-handed variety. So that's so the amazing thing is that this was discovered in 1880, and now we are 133 years later. Still, they haven't found a process that naturally just generates left-handed amino acids, not the right-handed. So that's actually, you see how much progress has been made in this research. They haven't been even discoveries from 1880, they haven't been able to explain that yet. So that's so that's the next stumbling block for the spontaneous origin of life. How do you get purified amino acids with only left-handed amino acids? Then again, if you jump and we assume somehow that there's the right kind of amino acids, there's only left-handed amino acids. But then how do you get them built into the right kind of chains, the polymers, as they're known? Again, amino acids, they bind in one particular combination. They can actually combine in several ways, but they only bind in, let's see here, they're called peptide or peptide, isn't it? Actual proteins in biologically relevant proteins, their amino acids are connected by peptide bindings, and they could bind in many other ways. So what natural process would make them only bind with peptide bindings? Again, we have not discovered any natural process that just causes peptide bindings amongst amino acids. So that's another something, no one knows how this could arise. Yeah. So these are just some examples to give an idea about the number of problems that this idea of origin of life being a spontaneous generation of amino acids and proteins. Actually, what we're talking about here is one particular approach to the origin of life, the chemical origin of life, which is that proteins came first and then the rest came after. But of course, there are actually all other theories also, which we'll just discuss now also. But so far it's been the idea about proteins first. And back in 1991, Stanley Miller, he stated, so okay, 1991 is 20 years back now, or 22 years back, but again it hasn't changed. Stanley Miller, he didn't change. Stanley Miller, he died in about 4 or 5 years ago, something like that. And but in 1991 he said the origin of life has turned out to be a much more difficult problem than I or most other had ever imagined. And that was after he said that after discussing many of these difficulties, which I'm trying to just give a hint of. Actually, he admitted that we had this one seemingly great success in '54, and since then we've just been discovering problems basically. So, but of course that's at the face of it, that's just one of the approaches to the origin of life, where proteins came first. Other scientists are proposing that it was some other elements that came first, and then later on proteins they arose. And one of those theories is that the RNA came first. As you may know, there are, of course, a number of chemical elements which are very important in living organisms. Proteins are one, DNA is another, RNA is also one, and then also there are RNA, and then I can explain a little, of course, what it is, and also there are sugar molecules, and there are fats or lipids, and they all they're all very important. But here, proteins, RNA and DNA are especially important right here. They're very much connected with each other. And in life as we know today, then DNA is where all the information which specifies the complexity of the cell. Of course, they say all the information, that should also be taken. Actually, it's not completely true, but let's still assume this is where all the information that specifies the complexity of the cell is stored. But DNA is actually just a passive molecule. It's not active. It's simply a molecule which stores information. It's a molecule where text is written on, basically. But otherwise, it's not active. It doesn't enter into any. It's not what they say is catalytic. It's not an active molecule. Then there are two active molecules. Proteins are very active, are involved in all kinds of reactions in the cell. Proteins, they act as, in many cases, as different kinds of machines, nano robots and like that, working inside cells, perform all kinds of functions. And they also are what is kind of just the structures, you might say, the beams and the building blocks that kind of build the cell are also made of proteins. And proteins are, they are involved in many kinds, but they're very active molecules. And then RNA is also a molecule that is very active. And RNA is also somewhat an intermediate between proteins and DNA, because the information on DNA is actually kind of read by the RNA, and then the information on RNA is being translated into the creation of proteins. I think we discussed that. It was in one of the first evenings we had, wasn't it? You remember that? No. You're also familiar how that functions. It's kind of, how the gene is a protein on a particular. There's information for the creation of a protein on DNA. That's a gene. It's kind of, today's kind of basic biochemistry. And so RNA is an active molecule. It can perform to some degree the same activities that proteins can. It's the RNA. I'm not sure you asked what it is. It actually means ribose nucleic acid. Ribose, and it's DNA. That means deoxygenated. Ribose nucleic acid. The difference is that ribose is a sugar molecule, and the difference between DNA and RNA is that a hydrogen molecule has been removed from the DNA, as far as I understand. They're somewhat similar in the structure. But RNA is an active, is a catalytic and active molecule, and therefore some scientists have proposed that RNA was actually the first molecule, not proteins, but RNA was the first molecule. And if you read a lot of the literature, they talk about the RNA world, and many have thought this is actually a solution to the origin of life. But again, as it turns out, there are similar problems like we discussed, that have the scenario of proteins first. Similarly, the problems you also find when it comes to RNA first. First of all, actually, ribose turns out to be a very difficult molecule. It's a fairly simple sugar molecule, but still it turns out it's practically impossible for ribose to generate spontaneously in any scenario, actually. So just to get this element has turned out to be difficult. But even if you had the same problem of assembly, spontaneous assembly is there. It's also both DNA and RNA also come in left-handed and right-handed varieties, like proteins does when they form naturally. But in living organisms, they only use, in this case, right-handed forms of these molecules. Again, how you can get them purified